Assalamu alaikum guys. Today we will discuss about gene and the different units of gene. First of all, we should know that what is gene. Gene is the polynucleotide chain. As we know that DNA is a polynucleotide chain controlling specific trait. The unit of, for example, this is the polynucleotide chain and we just take this part, so, and this part is going to control specific characteristic or trait of an organisms. So this part is called gene. Now this is the simple concept that uh, a single gene is responsible for controlling a specific trait. Single gene, single enzyme concept. And what is enzyme? It is basically a protein unit. Different scientists, for example, Beadle et al., they propose that one gene can make a single protein. And now we are going to explain this one gene and one protein concept. For example, we have DNA, and this DNA is going to produce a single molecule of messenger RNA, and this is translate and produce a amino acid chain, and this is called the polynucleotide chain, polypeptide chain. It has many and many amino acids, and when these amino acids combine and form a unit, this is called protein. So this is clear from this observation of Beetle and Titum that a single gene can produce a single protein. But in the recent studies and the recent researchers, it has been clear that there are many genes involved for production of single protein, and single gene is also involved to produce many and many proteins. So this one gene and one protein concept has changed, and we can also say that a single gene can produce a single polypeptide chain, but it is also possible that for a single polypeptide chain there are many genes involved for its production. Now we are going to discuss the units of the gene. So the gene has three units. There's cistron, recon, and mutant. The first unit, it was been coined by the Benzer, and he told us that DNA produce a messenger RNA, and the polypeptide chain, when translated, then it produced the protein. But this is not the case, because from the parents, when the genes or the chromosomes goes downwards, there another process which is called crossing over. So during crossover, these units or these genes change with each other. So it's not possible in all cases that the same progeny will be produced, means the same type of polypeptide chain will be changed. For this term there are two terms used: the wild type, which is original. For example, this is the chromosome, and these are the genes on the chromosome. But when there is a mutation or crossing over occur, after crossing over there is change. There are the XX genes. These are on the same location, but here the X has been the another X is changed by dot, and in this case the dot has been changed by cross. So the crossing were taken place and the wild type has been changed to mutant type. So the cistron, here we can define it's a functional gene, and the functional gene, it has the capability of the functional gene that it may have more than one types. For example, this is the gene, now it's of this type, and in next generation there is also the possibility that the type of this functional gene can be changed and polymorphism can be occur in the future progenies. The second unit, which is the recon. What is recon? Recon is the smallest unit, and when we say the smallest unit, it means that the smallest unit of polynucleotide chain. This is the polynucleotide chain, and we consider this is the smallest unit here. It is the recon. But what is the characteristics of recon? It can undergo recombination, means that there is chances that it will be changed from its original type to a new type, and this concept has been coined by Binzer in 1962. And he told us that there is the possibility of recombination in the DNA molecule when it proceed in the next generation, or during fertilization, during reproduction, during meiosis. This is also possibility, as we have already discussed during the crossing over. The genes change their position on the chromosome when they go to the next generation. Now, what's the recombination? How it can proceed? For example, we have a bacteria A, and this is the media, and when this bacteria grown or undergo reproduction and it produce progeny, it produced product A. Okay, but if we grow this bacteria here, it can produce the product B as well. So the question arises that how it can produce the product B, because this bacteria was responsible for production of product A and it has produced product B. So it is due to the mutation, as we have discussed here, that due to mutation, when the gene type or the nucleotide type change, so it can give rise to a new type of messenger RNA. Definitely, when the new type of messenger RNA produce, it will produce new type of polypeptide chain, and ultimately a new protein will be produced, as in this case we have mentioned here that the product B would be produced. Okay, the other part of the gene is the mutant. Mutant is the smallest unit of DNA or chromosome. As we know that chromosome is made up of two things. One is protein, and that protein is called histone, and another part is DNA. So the smallest unit of DNA or chromosome that undergoes mutation, the unit, the specific unit which have the capability of changing, which have the capability of mutation. The mutation may be intrinsic due to the internal environment of the organism, and the change is occurring. The one factor involved in the mutation is the crossing over, and the extinction factor, that may be the radiations and environmental changes that can also influence the structure of DNA, and ultimately their mutation occur, change in the amino acids. So here, as we have mentioned, that the smallest unit of DNA or chromosome that undergoes mutational changes, the smallest unit is the nucleotide. As we know that DNA is the polynucleotide chain. So the single unit of DNA is the nucleotide: adenine, thymine, cytosine, and guanine. So the mutation may be due to the nucleotide single nucleotide polymorphism, or there may be deletion and there may be insertion. The new type of nucleotide can also be mm, added to the polynucleotide chain, and there may be the replacement. Substitution can also occur during mutation. So the shortly, this strand is the largest part in the gene, and the followed by recon, and then the mutant is the smallest of all of them. But in some cases, the recon is also in the same size of mutant, but that should not be increased from one nucleotide. Hope you guys you have got the concept of the gene units, that the gene is made up of three units: cistron, recon, and mutant, and each unit has own specificity and its own characteristics. Thanks.